Hello everyone. In our today's video, we will be learning about vitamin C, mainly how to synthesize vitamin C from D-glucose. We will be learning about what is the importance of vitamin C and what its deficiency causes. So let us first try to see what vitamin C is. Vitamin C, also known as ascorbic acid, is a water-soluble vitamin. So remember that vitamin C is a water-soluble vitamin. That means it will not be stored in our body. It will be readily excreted by our body. So that means vitamin C has to be taken regularly in our diet. Okay. Vitamin C plays an important role in production of collagen in the body, which is a type of protein need for various tissues, including the skin, tendons, ligaments, blood vessels, bones, and cartilage. It is known for its role in the process of tissue growth and repair also. Okay, that is the importance of vitamin. That is the role what vitamin C plays in our body. If it is in deficiency, okay, if we have a deficiency of vitamin C, then it usually the effects of deficiency of vitamin C can be seen as we are actually growth is retarded, wound healing process is retarded, scurvy, okay, breaking down of immune systems, okay, spongy and bleeding gums, fragile blood vessels, okay, exhaustion, nervous breakdown, high fever. These are basically the effects of deficiency of vitamin C in our body. So as we can see, vitamin C will be very, very important for us. So now what we'll be trying to see is how to synthesize vitamin C from D-glucose. Okay. So the first step in the synthesis of vitamin C from D-glucose is hydrogenation of D-glucose to D-sorbitol with nickel as catalyst. So we'll do hydrogenation. I hope everyone can understand what is hydrogenation is. Hydrogenation is a process of addition of hydrogen. Okay, so how do we do it? We take the help of nickel, okay, as catalyst. That is very common. So this is your D-glucose, okay, and I hope everyone knows that glucose has a aldehyde group, okay. So this aldehyde get reduced, okay, with hydrogen and nickel as catalyst, that is undergoing hydrogenation to go for alcohol, a primary alcohol. So your aldehyde get reduced to primary alcohol, and this is known as D-sorbitol. Okay, then what do we do is in step 2, microbial oxidation or fermentation. So we take this sorbitol and ferment it, okay, to L-sorbose, okay, and with the help of this particular bacteria, gram-negative bacteria, which is nothing but Acetobacter suboxidans. Acetobacter suboxidans is a gram-negative bacteria. It is considered as a vinegar bacteria also, meaning it can produce vinegar. Okay. It is also a ketogenic. It converts sorbitol, okay, to 2-ketosorbitol, which is nothing but L-sorbose. Okay. So this is what happens. So you take D-sorbitol, okay. This D-sorbitol in the presence of Acetobacter suboxidans, okay, you can clearly see it removes 2 hydrogen, okay, and oxidize it. That is what, okay. So it oxidize it and convert into a keto group. You can see clearly, okay. So this is what actually is the L-sorbose. Then in step 3, so basically it is not step 3, but we are basically writing the furanose form of L-sorbose. Okay, so this is the furanose or the cyclic structure of L-sorbose, so that we can show clearly what happens in the next step. Okay, so this is the cyclic form, okay, of L-sorbose only, which we called as the furanose structure. Then the step 4, in which what do we do is we protect the 4-hydroxyl group of L-sorbose by formation of acetal with acetone and acid to diacetone sorbose. So this is our L-sorbose, okay. You can see that there are alcoholic group, okay. So this, and this, these are alcoholic groups, okay. So we react with acetone, okay, and I hope everyone knows that and alcohol reacts with ketone, okay, to form actually acetals, right? So that is what actually we are trying to do over here, okay? So it forms acetal. So this is the acetal which is forming a cyclic acetal. Over here also it forms the cyclic acetal, okay? This is 2-acetone which we are taking in presence of acid. And remember that in the process, this particular alcoholic group will remain as it is. And that is what we are trying to do. We are protecting this 4 alcoholic group. Now the question is protecting it from what? In our next step, what we'll be going to do is basically oxidize this to carboxylic acid, okay? So when we take oxidizing agent, what oxidizing agent we take is KMnO4, which is a very strong oxidizing agent, which will not only oxidize this, but oxidize the other carbo-alcoholic group also in the system. So to protect them, so first, what do we do is we convert them into acetal, okay? And then we oxidize this particular primary alcohol, okay? So that is what we'll be going to do in our 5th step. So oxidation with KMnO4 followed by heating with water gives 2-keto-L-gluconic acid. Okay, so we'll take this and then we'll oxidize. So this particular alcohol, okay, which we have again written in the simplest or in the open chain structure. So which you can see over here, this alcohol is converting into carboxylic acid and the other alcoholic groups are remaining as it is because we have protected it with the help of the acetone, okay, by forming it an acetal. Okay, so that is what actually why we have reacted it with acetone. Okay, so we get a carboxylic group. And finally, in step 6 is the cyclization. Okay, so what we do is this is the final step where we close or the ring closing step with the removal of water. So we remove the water, okay, and basically close the ring, okay, which connects the first carbon with the 4th carbon and forming a 5-membered ring, okay, which you can clearly see. It forms a 5-membered ring, okay. So this 5-membered ring over here, we can show it in another way, okay. Basically, it will lose water molecule, okay. So this same structure, this open structure can be drawn in this way, which is nothing but this, okay. So basically, it forms a furanose structure, okay. And this is nothing but your vitamin C. Okay, so that is how actually we prepare vitamin C from B glucose. Okay, so that is all about this particular video. So thank you, everyone.